Medicine Without Cruelty presents artificial intelligence. Now, artificial intelligence or AI refers to the simulation of human intelligence processes by computer systems. These processes include learning or acquiring information rules for using it, reasoning, so applying rules to reach approximate or definite conclusions, as well as the ability to self correct. AI encompasses various subfields such as machine learning, natural language processing, robotics, and computer vision, enabling machines to perform tasks that typically require human intelligence. AI is increasingly being used to develop human relevant, animal free methods in scientific research and testing that's accurate, effective, and reproducible. Now, AI is playing a transformative role in advancing non animal testing methodology by providing human relevant, efficient, cost effective alternatives. And it's got many applications. So, for example, toxicity prediction, machine learning algorithms analyze chemical structures and historical toxicology data, for example, to predict the toxic effects of new compounds. It can be involved in drug interaction modeling. So, AI models can simulate how drugs interact with target molecules, identifying potential side effects or toxic reactions without the need for animal models. Now, AI can also predict how a drug is metabolized in the human body, helping researchers understand its efficacy and safety. One of the applications of AI is the use of virtual patients and digital twins. Now, AI enables the creation of digital representations of the human physiology for testing purposes. It can provide a digital twin of organs like the liver, heart, or kidneys, which can be used to study organ specific drug effects. It can also provide full body digital twins to predict how a substance affects multiple organ systems, mimicking real world scenarios. It can also be used in personalized testing, so virtual patients based on real human data allow for individualized testing, such as evaluating a drug's effect on specific genetic conditions. Now, digital twins or DTs are created using machine learning models that are trained on patient data from clinical trials and observational studies. They can represent a range of systems from individual cells to entire human beings. And they can be used in drug development to accelerate the process of discovering and developing new drugs. It can be used to model diseases such as identifying drug targets and disease biomarkers, and can also be used to test new compounds and simulate how they might work on disease pathways. And when it comes to drug development, it can improve disease understanding, so it can help researchers better understand diseases. It can also help researchers identify disease biomarkers. So, as you know, there are certain hallmarks of diseases. So, we can use this digital twin to help identify these hallmarks. It can also accelerate drug development because it can help speed up the process of drug discovery and development, and it can also help improve product quality. So it can help manufacturers monitor the quality of their products throughout the manufacturing process. Another application of AI is the use of high throughput screening. So it can enhance the efficiency of these high throughput screening or HTS platforms by automating data analysis. It can process data from cell based assays to identify promising drug candidates. Its algorithms can analyze large data sets to quickly determine the safety and efficacy of chemicals. And also, AI powered image analysis of cell cultures identifies subtle changes, so perhaps a cell morphology or other classical hallmarks that indicate either toxicity or its effectiveness. Another application, and this is what I mentioned in the previous podcast about organoids and organ on a chip, but AI can empower and advance these 3D culture systems. For example, predictive analytics. AI can process data from organ on chip systems to predict how human tissues will respond to drugs or toxins. It can also use machine learning algorithms to analyze real time data from organoids, such as changes in morphology or function. And also, because we can use interconnected organ on a chip systems to simulate full body responses, AI can then integrate data from these multi organ on a chips. Its applications are also in regulatory toxicology. Now, I'll talk about the Tox 21 initiative in the next segment of this, but AI is a critical part of the Tox 21 program. I'll discuss it in the Omics podcast. It also can be used to assess the safety of cosmetics, chemicals, pharmaceuticals in compliance with regulatory guidance. And with it can ensure consistency and reproducibility, which is a key requirement in regulatory toxicology acceptance. AI can predict the safety of chemicals by understanding and analyzing their structures and biological activity. So, for example, with read across methods, AI can infer the toxicological properties of untested chemicals by comparing them to structurally similar compounds with known properties. And it can also predict the impact of pollutants on human health and ecosystems, offering alternatives to traditional animal based environmental testing. It can be used in rare disease research and personalized medicine. Now, AI can use patient data to model diseases in silico, so using computational methods, bypassing the need or the necessity for animal models because it's not necessary. AI can identify existing drugs that might be effective for rare disease by analyzing molecular interactions and disease pathways. It can be used within personalized medicine approaches. For example, genetic profiling, AI models can predict how individuals with specific genetic profiles can respond to drugs. And also with personalized patient derived organoids, AI can analyze that data to tailor treatments for cancers and other diseases. I spoke a little bit earlier about AI powered imaging systems, but it's really incredible how they can analyze cellular changes in response to drugs or certain chemicals, toxins. For example, AI can identify subtle changes that, say, maybe even a human might not even be able to see in cell shape and structure, indicating whether a potential drug is a toxin or it's effective. It can also provide phenotypic or behavioral or characteristic screening. So machine learning extracts patterns from imaging data to predict biological outcomes, because if it sees particular patterns within the imaging data that's either positive or negative, indicating either toxicity or drug efficacy, then we know what the biological outcome will be. And also with automated scoring, because if you are, say for example, you are looking at a vast population size, now AI can score those cellular responses to substances, thereby eliminating human bias and massively reducing the time needed. Now, thank you for listening.